Post-transplantation cyclophosphamide has really made a dramatic impact on the field over the last several years and is increasingly being used as standard of care, not just for haploidentical transplantation, but also for matched donor transplantation as well. But when we take a step back and look how we got to this stage, it's important to recognize that how we give post-transplantation cyclophosphamide did not come through sort of prospective analysis of different dosing schedules and timing of the drug. This really came largely from extrapolation from murine skin grafting models, in which it was thought that you needed a very high dose of the cyclophosphamide at 200 milligrams per kilogram, and that you needed to give it on days 2 or 3 after transplant. And so when this was taken to bone marrow transplantation, in the first clinical trial, they said, okay, well, we think we need a very high dose of the drug. Well, 200 milligrams per kilogram you really can't give to a human. You can give 50 to 60 milligrams per kilogram. And we've had some success of treatment of aplastic anemia with 50 milligrams per kilogram. Let's go with that. And days 2 or 3, well, day 3 is a little further away from the conditioning regimen, and so that may possibly decrease the risk of toxicity of the treatment. So it was chosen to give a dose of 50 milligrams per kilogram on day 3. And that's what was done for the first phase 1 clinical trial that began in 1999 at Johns Hopkins. With that publication in 2002, there was positive results here, and now being able to safely do T-cell replete haploidentical transplantation. But there was still a substantial risk of graft-versus-host disease. And so in the phase 2 study, there was run jointly between Hopkins and Fred Hutch Cancer Research Center. And in the Hopkins patients, they decided to add a second dose, which they empirically added on day 4. And so, whereas the Hutch patients received just the single dose of 50 milligrams per kilogram on day 3 only. While there were no differences in acute GvHD between the two different groups, in this cross-institutional comparison of the outcomes of the patients, 40 patients treated at Hopkins and the 28 patients treated at the Hutch, there seemed to be a suggestion for less chronic, extensive graft-versus-host disease in the Hopkins patients who had received the 2 doses of the drug. And so that's really how we got to what's the current clinical standard of 50 milligrams per kilogram per day on both days 3 and day 4. And this has not been rigorously tested prospectively as to whether that's the best dosing schedule. We have done extensive mouse modeling and haploidentical transplantation models in my lab, and found actually that in hematopoietic cell transplantation, intermediate rather than very high dosing of the cyclophosphamide seems to actually produce better prevention of graft-versus-host disease and decreased mortality in our models. And this is associated not just with survival outcomes, but also reduced histopathologic as well as clinical graft-versus-host disease, and also actually preferential regulatory T-cell recovery, which may in part explain the better prevention of graft-versus-host disease. We also studied the different timing of cyclophosphamide and did find that the days 3 and 4 is among the best dosing schedules, but that the maximal efficacy of the drug is actually at day 4, not the day 2 or 3 as was thought to be in the skin grafting model setting. And so this motivated us to take these 2 major findings on potentially intermediate dose of cyclophosphamide, as well as the later maximal efficacy of the drug to a phase 1 study, or phase 1/2 study at the National Cancer Institute, which we fully enrolled, which sought to de-escalate the exposure of post-transplantation cyclophosphamide for myeloablative haploidentical bone marrow transplant. So the first 5 patients in the study received the standard dose of the drug, so the 50 milligrams per kilogram per day on days 3 and 4, with 2 experimental dose levels. One was the half dose, the dose that was shown to be the best in our hematopoietic cell transplantation models in mice, of the 25 milligrams per kilogram per day, on days 3 and 4 for the first experimental dose level, and for the second experimental dose level, that same half dose, but on day 4 only. We found that both of those altered dosing schedules were associated with good protection against graft-versus-host disease. In fact, at the dose schedule in which we gave the half dose on both days 3 and 4, there were no cases not just of severe acute graft-versus-host disease, but there's no cases actually of even grade 2 to 4, what would be considered to be clinically significant acute graft-versus-host disease, suggesting that we could get excellent control with less cyclophosphamide dosing. Not only that, but with that lower dosing, we saw 5-day faster neutrophil engraftment, 11-day faster platelet engraftment with decreased transfusional requirements, as well as less toxicity of transplant, so less severe, shorter duration of mucositis, and suggestion potentially of better functional immune reconstitution, as indicated by decreasing severity and duration of BK virus-associated hemorrhagic cystitis. And so we're excited by these findings. We have 2 other clinical trials, one that's been enrolling for about a year and the other that's about to start enrolling, testing whether this reduced dosing of cyclophosphamide may also apply to other transplantation settings like reduced intensity conditioning regimens, whether using peripheral blood stem cells as the donor source rather than bone marrow, or using HLA-matched donors. And it is possible that the optimal dosing of cyclophosphamide may vary depending on the different transplantation schema.